Thought maybe you needed a break, and then also it'll help us to sort of pick up our pace a little bit and get back on schedule. So we should be able to finish that off on Thursday, I'm hoping. Lab, we will have lab for once. If you're curious, the houseflies never emerge, so I'm pretty much convinced they're dead. I don't know what else could have happened. So we may try again later in the semester. But we have birds that are coming to feeders that I set up down by Lake St. Francis. I set them up last week. I was there on Saturday, and they were just there were chickadees and nuthatches that just didn't seem to care about me being there restocking the feeders. So I think we'll hopefully have good attendance in terms of birds at feeders, even though it'll be in the afternoon. I did just briefly look at the weather forecast, and it didn't look great, but it didn't look bad. So Thursday, it does say PM thunderstorms, but if you look and see, I like Weather Underground because I think this is just beautiful visualization. But it looks like the worst starts around 4-ish, which is when our lab is over. So I'm hoping that we are able to get in before it really starts to come down. If it starts pouring when we're out there, I don't know, I guess we'll have to call it, but we will hope it does not do that. And we wouldn't be calling it because we're getting wet. We'd be calling it because the birds won't do anything if it's pouring down rain. But if it's just sort of sprinkling or just sort of wet, it'll be fine. So I do recommend that you bring some manner of sit-upon. So if you were a Girl Scout or are a Girl Scout, probably too late to be a Girl Scout. But anyway, if you were a Girl Scout, you're aware of what a sit-upon is. This is a device upon which you sit. And the thing about that is that it hopefully will be waterproof or water resistant of some kind. I find a towel wrapped in a trash bag works very well, and that makes for a much more comfortable sitting environment than sitting on wet ground where your bottom is getting soaked. So I also recommend bringing some manner of appropriate clothing depending on what the temperature and weather and such conditions are. If you want to bring a poncho or something, if you're worried about getting wet, then you should do that. Hats are nice. I find, I can get wet, but if I have a hat to keep the rain out of my face, I'm so much happier. The world is better in that case. So any questions about anything right now? All good, all happy? Exam's over, so that's nice. Did it go okay? I haven't looked at any yet, but getting the blank stares of, oh my God, look. So, okay, well, hopefully it went all right. Get on them as soon as I can. I think what I remember is that we were going through the different axes, and we had just barely gotten to this one. So we had talked about the HPG, hypothalamo pituitary gonadal axis, and I think we had just started to talk about the hypothalamic pituitary adrenal axis, or the HPA axis. I thought just to get our brains up to speed, I'd sort of re-go over this slide. So the idea with this particular axis is that you have signals that originate in the hypothalamus, and the hypothalamus is a part of your brain that's getting information from other parts of your brain about what's going on. It could be our brains, it could be the brains of some mammal that we're studying, or some bird. But they're receiving information about what's going on in the world around you. And so if an individual is encountering stress, and this could be social stress, this could be tax evasion stress, this could be also, food stress, so there's no food available in my environment. Or this could be, there's predators about and I have been chased recently. Whatever the source of the stress is, a typical response to stress is that the hypothalamus releases corticotropin-releasing hormone, or CRH. It is a releasing hormone, and all the releasing hormones, their jobs are to tell the pituitary gland to release a different hormone. And in this case, the pituitary releases adrenocorticotropic hormone. The term is terrible in a way, but on the other hand, it looks terrible and long, but at the same time, it actually is really descriptive. So adrenocortico refers to the fact that it's going to the adrenal gland, and in fact, it's going to the cortex of the adrenal gland. So adrenocortico refers to the destination of the thing, and tropic just means changing. So it's a hormone that's going to the adrenal cortex and causing a change there. And the change that it causes is that it causes the cortex of the adrenal glands to secrete glucocorticoids, which are sort of a general class of hormone that results in stress responses. So we consider these things to be stress hormones. In humans, it's mostly cortisol. In other animals, it'll be some other manner of glucocorticoid, but they all kind of do the same thing. And that is that they activate responses that allow animals to handle stress and to cope and deal with stress when they're encountering it. I tend to think that they work best when you're dealing with acute stressors. So these are short-term stressors, short-term stressful events that are happening over the course of an hour or a day, maybe a week or so. Where you start to run into problems is when you encounter chronic stressors, where you have extended stimuli that last for longer than that, and then that sometimes these stress responses can result in damaging effects on the individual. So for example, what do they do? So for example, with acute stressors, glucocorticoids will activate behaviors or physiological responses, it should also say up there, that allow you to adjust to stress. So you'll see changes in the behavior of the animal. You'll also see changes in terms of releases of energy from metabolic stores. So that can mean you're breaking down fat stores, for example, and liberating those so that you have now more energy molecules available in the blood that cells can use in order to power processes. So, you know, if you are encountering predator stress, you may increase your metabolic rate, and you also will see increased energy that's available from these metabolic stores. Similarly, if you have food stress and there's no food around, you can release fuel from your fat stores, and that can then help to supply you through that stressful time. They can result in stimulation of your immune system so that you're actually upregulating your immune system to deal with some kind of an immune challenge as you're dealing with stress. It can also result in suppression of reproduction. So the idea here is that in a time when you're encountering substantial stress, that is probably not the best time to be routing substantial amounts of energy into egg production, for example. And in many animals, that's a major energy sink, is production of eggs, at least within the females. So if you suppress that for a time, you can save all that energy and channel it into physical activity, or channel it into, you know, just maintenance activity while you're dealing with the fact that there's no food around. So this is what it's for. Like, these are the advantages of the system. There are certainly costs to it. So if you encounter chronic stress that's going on for an extended period of time, it can result in substantial depletion of your energy stores. It can ultimately actually impair immune function, and the mechanism there isn't clear until you really get into studying the thing. But you know, you're kind of upregulating for a time, but then you could think of it as sort of exhausting the immune system as a result of chronic, prolonged stress that you're encountering. The bottom there, it just sort of describes some examples, and your book goes into these in more detail, but you know, you can think of examples of stressors can include food limitations, social conflict, adverse weather conditions, and these could be acute stressors, but they could also be extended, prolonged stressors. You do see examples in many situations, and we'll talk about these later in the semester, where you see in social species, you'll see that subordinate females within social cooperative breeders, a lot of times the subordinates are not just sort of subordinates, and you know, I mean, they're subordinates. They're not just sort of happily going about their duties in this harmonious relationship. Oftentimes they're actually stressed by the dominant females within an environment. So for example, if you look at naked mole rats, are a classic example of this. There's like a dominant female queen, and she basically just bullies and beats up on all the other females that are present within her nest. And as a result of that, those females, in part due to their stress response, do not reproduce. And so the queen is the only one reproducing within the colony. And the other ones are not reproducing because they're basically stressed out all the time. And you know, as a result, you have a relationship that's kind of like what we saw with the honeybees, where there's a dominant queen that's doing all the reproducing and the workers really don't reproduce. In naked mole rats, there are, if you go and monitor glucocorticoid levels as an indication of how stressed out the animals are, you see substantially more glucocorticoids in the workers than in the queen. Similarly, if you look at macaques, these are monkeys, and you look at those individuals that have more antagonistic relationships and then compare those to individuals that have more friendly relationships with the individuals around them, those that are involved in more antagonistic relationships tend to have higher glucocorticoid levels because, again, that's an indication that they are more stressed, whereas those that are in more harmonious, friendly relationships tend to have lower stress levels. So you can use this as sort of an indication of how stressed an individual is. And in fact, that is often what people do. I know Dr. Wolf has long been talking about doing, she may have even done this once, and I never saw the results of it. You can do a spit test for cortisol levels in people. So you basically have them spit in a cup, and then you can assay the cortisol levels. And she wanted to do, like, go find people right before they're about to take a anatomy, physiology, or comparative anatomy lab exam, and then assess them before, and then assess them, like, 5 days later, under less stressful conditions, just to measure the cort levels. And a lot of times we do abbreviate. We call it CORT. So CORT has to do with glucocorticoid or cortisol. Anyway, that's sort of the story on those guys. Any questions on glucocorticoids? I think a point I would make, I probably didn't make it. One of the cool things about this investigation of hormones, and a thing that I don't think I really grasped for quite a while, is that there are commonalities in how virtually all vertebrates respond to stress. And so we can use measures and assessments of glucocorticoids as a means, sort of a ubiquitous way to assess how different animals respond to stress. Similarly, many animals respond in very similar ways. I mean, there's definitely variation, but similar ways to sex hormones like testosterone and estradiol. And so we can use these as measures that apply across multiple species. They're sort of a generalized measure of reproductive function or stress. And so in that way, they can be a really nice tool that we can use to assess sort of the state of a particular animal. All right. So the first one, I'm always interested to see how people do with this one. So this one, I'm asking you to look at these diagrams and read the text, try to figure out exactly how it is that this PER protein works, because this is ultimately at the heart of how we have some kind of a circadian rhythm. Is we have these little molecules that increase and decrease and change their concentrations through this process, and the figure tries to show how this might work. So I thought maybe I would just like point as you talked. I think you got this one right. So see what you got, and then we'll fill in details if there are more to fill. That's fine. That's why I'm going to point instead of try to type everything.
So I have that gene known for the protein BER, which will rapidly build up on the inside and outside the cell. Two other genes code for an enzyme, ch6, which helps to break down the red blood cell. When BER proteins bound together with other complexes, it's TIM, it's not broken down quickly. Then more intact BER is taken back into the nucleus where it lost activity for the.
That was not that much. I thought it was going to be like 5 more minutes of.
I don't know. I thought it was kind of all over the place.
To me, it sounded pretty good. I mean, anybody? I mean, I could try to describe it too. I mean, I actually thought you had a very nice description. So, like, this is my understanding. I mean, they're showing mammals. They're showing flies. They're pretty similar in a lot of ways. I don't know, they show some different details in one place versus the other, but the basic idea is PER is manufactured, so PER is a gene, so it's part of the DNA. It's manufactured here, so we're transcribing into messenger RNA, and then we're translating into PER, the protein, capital PER. And then as you said, PER can be broken down by this guy, the CK, I don't know if that's an LE or IE, I honestly don't know. Some kind of a kinase or something. So this will degrade this protein. Here it is being phosphorylated. And at that point, it's degraded, so it's non-functional at this point. However, PER can also be protected by glomming onto other stuff, including this TIM protein. And so if it's protected by TIM, then it will come in, and PER will, when it builds to high enough levels, it will re-enter into the nucleus and it will actually bind to and inhibit the DNA here, or interface with the DNA such that it prevents additional PER from being constructed. And so the idea is that initially you'll make PER, and a fair bit of it will be captured with TIM, and so you'll be able to sort of gradually accumulate PER over time. I always like to draw a graph. So if you measured your concentrations of PER over the course of a 24-hour period, it would kind of start really low, but as you produce it and build it, it would increase in concentration. And that would continue until such time that you kind of run out of TIM. Here it's actually showing in the fly one anyway. They've got good indications that you'll actually inhibit production of TIM when this TIM-PER complex re-enters into the nucleus. I don't know if they don't have that indication in mammals or what, but it's the same basic idea. So as you kind of build up to very high levels of PER, it's protected for a time, but then eventually what happens is you kind of overwhelm TIM's ability to protect it. You manufacture a bunch of this kinase, and so the result is that the concentrations start to drop again. Well, I should say it's dropping for two reasons. One is that the kinase is breaking it down. So here's, it's showing, here's the kinase breaking it down outside the nucleus. It's breaking it down inside the nucleus. And so that is going to take a toll on your PER concentration. And at the same time that the kinase gets going, PER has also been inhibiting its own production. So once you get to good levels of PER, PER shuts down its own production right here. It's showing inhibition right there. And so as a result, you're not doing this step anymore. You're not making more PER. So we're kind of, we build up PER for a while, and then we inhibit further PER production. And we kind of overwhelm Tim. I have an Uncle Tim, so I always sort of visualize him somehow trying to protect this thing. And we're overwhelming Tim. And so at this point now, we can't protect our PER from the kinase, and we're not making any more of it, so its concentration will drop again. And this will take, for this whole process to go up and then go down, this takes almost exactly 24 hours. Does that make sense? So I don't need you to be able to redraw this whole thing, but I do want you to understand this idea that we're kind of building up PER, and then it sort of self-inhibits itself. It's a feedback inhibition process. And so at that point then, the concentration will crash. And it does this every single day. In fact, I think, if I remember for PER, I think this is sort of like at the peak of the night is more or less when that is happening. I think that's right. And so this would be kind of like, at noon, well, I don't know, maybe it's like 2 or 3 in the afternoon when we're at our lowest PER levels. It kind of doesn't matter as long as, it's cyclical and it's happening on roughly a 24-hour period. And then we can train this. There's signals that can affect the, where exactly we are in this to make sure that we're calibrating it to the day-night cycles and that kind of thing. But this is our basic biological clock that we're seeing happening in the suprachiasmatic nucleus. Is that okay? Questions? That's what we're shooting for. Get the people up. So number 2 is kind of an extension of that. So this was PER in a mammal. I guess they also showed in a fly. This question's asking, how is it that this particular figure is demonstrating that PER is involved in fruit flies as well as we saw it already in the mammals. So I don't know, what'd you put for this guy? And it so we've got, here's the gene, and it does show these 3 different mutants. So what are these different mutants? Did you see what they're showing in terms of the long mutant versus the short mutant versus the rhythmic mutant? Do you know what? I'm putting you on the spot here, but Yeah, that's pretty much what it is. So you've got here the wild type, that's our normal. So wild type is the type that if you went out in the wild and you caught a Drosophila fruit fly buzzing around, that's what it would be. And you see that they have a nice sort of steady rhythm. I don't know if this is, I forget if this is meant to be when they're sleeping or when they're awake, but the idea is that they're very steady and rhythmic every day. You have a long period mutant where they seem to be, this period here is a little bit longer in every single cycle. So instead of having sort of a normal day-night cycle, a normal 24-hour rhythm, they kind of have a longer period of activity here, if this is activity. Similarly, you've got the short period mutant, which is down here. They have shorter periods of activity down here. So you've got what's happened here is that at each of these locations, there's basically just a small variation that's occurred, but those variations are driving changes in the circadian rhythms because we've changed the PER gene. The PER gene doesn't work like it normally would, and as a result, you have differences in the behavior. My favorite is the arrhythmic one, where this mutation, this is probably like a nonsense mutation or something, this just blows the whole thing up. And so as a result, the animal just is, there's like no rhythm at all from day to day. It's awake and asleep at different times every day. There's no rhythm. It's just total randomness and chaos. That's pretty neat. Okay, so PER gene matters. One more thing, sort of related to that. So while the PER gene and its protein may be the basis of the biological clock and the suprachiasmatic nucleus, other molecules are also involved. So the first bit, 3A, is asking what is the major hormone that's used to communicate the suprachiasmatic nucleus's information about time around the brain? And there's something about jet lag and popping some pills. And so, Ava, I think this one is yours. What did you get?
I had melatonin is the major hormone used to communicate, and the direct consumption of this compound at night can help reset the sleep cycle.
Agree, disagrees? Anybody ever tried melatonin? I haven't tried it. You guys have? Does it work? Did it? After you take it? Yeah. Yeah. Didn't feel rested or something in the morning, or at least getting out of bed was hard. Yeah. My wife has tried it. I haven't tried it. My wife has more sleeping problems than I do. I fortunately, somehow, despite the coffee, seem to have a pretty consistent sleep cycle. Yeah, I mean, this is, I mean, you can just go into the store, into GMC or whatever, and you can just buy melatonin. And I've always been a little skeptical that it can pass through a digestive system and get into our system, but there seems to be good indication that it does do this and does seem to work a little bit. So if, the idea is that PER is this gene and this protein that's manufactured within the suprachiasmatic nucleus. It sends signals to the pineal gland, which is a little bit north and up in the brain, but then the pineal gland broadcasts melatonin, and it has maximal broadcast of melatonin as you're getting tired. And so that's why if you take melatonin at that same time, you're sort of basically duping your brain into thinking, okay, I'm getting melatonin surge, must be time to get ready and go to bed. And hopefully, if you are jet lagging, this may allow you to sort of reset your system more quickly. The next bit is asking about this PK2 gene, I guess it is, which I had never heard of until I saw this question. This is new for this edition of the book, too. I'm going to make this bigger so we can actually see it. But it says, what was the experiment that was done in this study, and then what does this tell us about function of PK2? So this one went to Megan. Yeah, so in what, so this is not the injection study that was mentioned here though, right? So this is the one where they're manipulating the light environment around the animal. So they've got a normal light dark cycle. They've got 2 days in, I don't know what DD stands for. Darkness. Yeah, right. So it's dark as opposed to light dark. Okay, that makes sense. Yeah. 2 days in total darkness and 8 days in total darkness. And so, like, what is the fact that you get peaks at the same time in all 3 groups and then a trough here and then a peak? What is that? What specifically is that telling us? I mean, I think you kind of hinted at it, but I want to just make sure we spell it out. Yeah, so the green and the blue one, these guys don't have any external cues. This is like these experiments we were talking about last time. These guys don't have any external cues, and yet they still are following the same kind of a rhythm in terms of expression of PK2 as the light-dark individuals were. So they're, at the same time of day, despite the fact that there's no environmental cue, they are still expressing this at a high level during this period of time, and then from hours 13 to 22, they express virtually none of it, and then around hour 22, they start expressing it again, just like we see in the individuals that were on the light-dark cycle. And so that at least is indicative that the gene is responding to a circadian rhythm. And then the thing that was sort of the clincher, that this really is sort of a driver of circadian rhythms, is that if you manipulate it, you can alter the sleep-wake cycle of the individual too. So, I mean, there's lots of genes and lots of proteins and hormones and stuff that fluctuate over the course of a day, and because they're responding to the signals about circadian rhythm. So, just the fact that it fluctuates doesn't necessarily mean that it's involved in determining your rhythm. It may just be responding to the rhythm. But the fact that then, as you said, if you manipulate it, that demonstrates that it seems to be important in terms of sleep-wake cycle determinants. So the point of all this is that the PER gene is not the only gene that matters, and even melatonin is not the only hormone that necessarily matters. There's at least 2 other genes that I've heard of that are circadian rhythm genes for humans. So that one of them is just called clock, which is nice and convenient. I forget what the other one's called, but there's another one that is mentioned in my neuroscience book. So there's a lot of these things, but they're all kind of working in concert. And a lot of times you have these sort of redundant systems in biology to make sure if one goes haywire, the other ones are probably working. And that's probably why we have all these sort of multiple systems going. All right, let's do 4. So 4 says internal clocks, be they circadian or circannual, require some machinery. Why should we bother doing them? So like, what might be the adaptive benefits to using them compared to just using environmental cues? So this one went to Tyler. What'd you get?
We're on number
4? Yep.
I put that it helps the body direct what needs to be done, or what needs to be produced, so that nothing is wasted in terms of energy and hormones. So like, for example, you don't exactly need to produce things that would help you with mating if you're active during the day, but it's nighttime.
Mm-hmm.
So that's what I was kind of thinking there.
Yeah, I'm just going to add a little ditty. So like if you have a process, so you mentioned mating, so there may be sort of a ramp up process or you may need to, it might not just be like a switch. You can instantly turn some certain behaviors on. Maybe you need to ramp up production of a hormone over the course of a day, or probably works better to think about it over the course of a year. So if you want to be ready to mate at a certain time, you can't wait for that signal, the environmental cue necessarily to happen, because you need to be ready by the time that signal happens. So it allows for you to sort of predictably, anticipating appropriate times for behaviors or changes in physiology output. Like that. You may have other ideas or explanations for this. I think there are probably multiple that you could put out.
Kind of going off that, I
species that are necessary for survival. So like in people, like we have to sleep, or like in the squirrels, like they have to hibernate, otherwise they'll like freeze to death.
Mm-hmm. So they're necessary for survival, so they need to have some kind of a reliable signal. Is that the?
Yeah, like a reliable like clock.
Yeah, so I like the idea. So the hibernating animal, you could sort of imagine if you had a really warm fall and early winter, and you don't have any kind of a circannual rhythm, the animal might not be getting environmental cues that are indicating it's about to be like horrible winter time. And so the critter's just sort of chomming around, hanging out, being lazy, not going through its normal stashing, not building up fat stores, and then all of a sudden winter hits, and it might be too late to make the adjustments you need if you're just relying on environmental cues. So having an internal system may allow you to have a more reliable kind of a clock as you're anticipating these kinds of changes. So the idea here is that environmental cues may be unreliable. I don't know. I'm losing my my words, but yeah. So environmental cues may be unreliable. So it helps to have a biological clock system so you can anticipate, or so you can have sort of a backup signal to oneself to make sure that you're doing the things you need to do at the appropriate times. To me, it kind of almost makes more sense at a circannual level than it does on a circadian level. I mean, I always sort of thought, can't you just look out the window and see if it's light? And if it's light, then you just go. But you could sort of imagine some circumstances where, if you're entering into dark environments, so if you're going into a forest or if it's particularly cloudy on a particular day, that could upset your rhythm if you're strictly relying on environmental cues, depending on exactly how it is that you calibrate those cues. Whereas if you have a biological clock, you can compensate and anticipate that those environmental cues may not always be working and reliable. Does that make sense? Virtually every organism out there has some kind of a biological clock. This includes plants, it includes fungi, it includes animals, it includes bacteria. So this is sort of a fundamental thing that seems to be advantageous for virtually all organisms to have some kind of a rhythm. The only places where you don't really need a rhythm are in a situation where there's just no fluctuation in your environment. So I actually never really went and checked, but I would anticipate, for example, that cave-dwelling species may not have much of a biological rhythm. I don't know that, though. That actually, somebody's got to have done that at some point. But because you might not expect that, because in cave-dwelling species, they just don't have any change in environment from day to day, and really even from month to month. So that would be a cool thing to look up. I'm going to write that down. All right. Just thought came up. If not, maybe that's an undergrad research project. Okay. The last one is on ELISA. This is a new bit that's in this book for this semester as well. But certainly ELISA, we have ELISA systems within our department. That was one of the first things Dr. Wolf did when she got here, wrote a grant and purchased one of those. So it says, provide a short summary of the methodology behind what your book calls a basic ELISA. And I thought, again, I would just sort of flip to the figure, if I can get there quickly. It's way up here. Thought that was it. Wait, here it is. Sorry, who did this go to? Alexandria, maybe? Yeah. Mm-hmm. Yep. Anybody ever get to play with one of these things or do something similar? You did? Summer? Okay. Okay. Yeah, I knew you did the Summer Academy thing too. So this is, I mean, there's a lot of techniques that are similar to this. So there's different kinds of antibody staining methods. This is effectively, it's kind of one. So the key thing is you have a particular antigen that you're looking for, and you have to buy antibodies that match that antigen. So this is, and those can be manufactured. So you just kind of buy this oftentimes in effectively a kit. But the idea is you've got little wells that have that particular antibody that is specific to the antigen that you're looking for. So if you were looking for one particular protein, you want to see how that protein is being manufactured. Maybe you're interested in melatonin, and you want to find out, is melatonin being produced? What you could do is buy an antibody that binds melatonin. So if you then add your tissue sample to the well, the melatonin will bind to that antibody. Then, like you said, you kind of wash it out. But at that point, like, that's happened, but you can't tell. Like, you can't see anything. So the whole point of having the second antibody system is that this is kind of your labeling system. You have a second antibody that swoops in, binds up here, and then this enzyme that's attached to your antibody can convert something that's colored into a different color, and then you just sort of measure that with a spectrophotometer. So you measure the change in color. So it's a nice, elegant little way to do this. I've seen other methods, like if you look at some of the other antibody-based staining mechanisms, they almost seem easier, where you basically just have an antigen that's sort of sitting in your tissue, and you apply the antibody, and then you apply a second antibody that binds to the first antibody that is colored or fluorescent or something. So again, the idea is that the second antibody that you're adding here is what allows you to actually see if the interesting thing has happened. The interesting thing was, do I have that antigen there at all? Did it bind to my first antibody? So the whole point of the second one is just to produce color, so you can perceive it. Okay. So then let's do B. And part B was broken up into parts A and B. So Sean, it says, assume you plotted the standard curve shown in figure B using standards. So that's why it's a standard curve. You have two unknown samples. One has an absorbance of 0.35, the other one 0.55. What would the concentration of the hormone be in those cases? What did you get?
For unknown substance 1, there would be a concentration of 6 nanograms per milliliter. And for unknown substance 2, I said about 11.8 nanograms per milliliter.
0.8. Very precise. Yeah, all right. Is that about where you guys are? I didn't actually do this, so I'd have to so 0.35 would be here. So that would be right around 6. 0.55 would be here, which would be, 11.8, I think exactly. Yeah. This is the idea, though, is that you've created this standard curve. This is, very typical. So we know when you produce the standard curve what the concentrations are of the hormones, and then you measure the absorbances. And then once you have that curve, you can take any absorbance and then infer back what the concentration would have been. And ideally, they're linear, which is what this one is. If they're not linear, then you need to be accounting for the fact that it's not linear. So if there's a curve, for example, as opposed to just a straight line through the thing. And then this last question is, what would happen, or what would you do, I guess, if your standard curve did not match, if your unknown had a reading that was outside the bounds of what your standard curve were. So I don't know, what did you put for this one, Melissa? I misspelled this. Did I put anything different for either? Well, we already asked about A. I kind of read this one differently for B. I think to me, I was reading it more of sort of like a practical, from a practical standpoint, what do we do from a method standpoint if we're sitting there and we have, so if we measure something and we get an absorbance of 0.25 or even 0.2 on our measurement. So that would be, that would be like here, or if we expanded the axis down a little bit, it might even be like down here. What if we were sort of in this realm? What would we be comfortable doing? I mean, what would you say at that point? I mean, what do you think? So you sort of extrapolate down. Yeah, so like, so this is, you have a nice equation for it. You can calculate a nice linear equation here. You could plug in the numbers and you could get an estimate of what the concentration of the hormone would be. And I think you could have an estimate from that. I mean, it would be low, but it would give you a concentration that would be like a non-zero value. I think some would argue that really you maybe shouldn't, though. And so if what we know from the standard curve is that within the bounds of a concentration of the hormone from 4 nanograms per mil to 14 nanograms per mil, we know there's a pretty linear relationship there. I think most people that were doing this would say that if you got a unknown value that was beyond the scope of that curve, so if your unknown was way over here, or if it was way up here, we don't really know the relationship there between concentration and absorbance, and it could become nonlinear out here or out here. We could have some sort of fluctuation in the curve beyond the point that we actually measured. So I think the argument would be, at least from some quarters, and this is sort of how I was interpreting it, that really you would need to have a different standard curve. You would need to reproduce the standard curve such that we get concentrations that are down in that range, so that we know it is linear throughout, and you don't have some sort of wild fluctuations. Because it could be like that this like plateaus off somehow, right? So maybe absorbance really never goes beyond 0.7, and so at that point then you can't tell the concentration once it's out here. So if you got an absorbance of 0.7, maybe it could mean the concentration could be anywhere from 16 to 30, and maybe you can't tell at that point. So it's important to have a standard curve that that bounds all of your data that you're actually trying to interpret. Does that make sense? I think that's what they were going for, as opposed to more of like a variation across species kind of an argument. Any questions on anything? Are we good with these? So we've got 12 minutes, so I do want to start into chapter 6, which is our next order of business here. So again, I posted these last night. If you didn't get a chance to print them, you can just sort of take notes as if you're going to print them and then transfer them over. Chapter 6 is a little bit of a departure from what we've been doing. Get that projected up here. We had a chapter that was sort of wholly focused on nervous system. We had a chapter that was on, nervous system, but also physiology and endocrinology. It was all very proximate in terms of its approach and its focus. And that is kind of how the book is constructed. So we kind of start proximate, looking at immediate mechanisms that are going on within individuals and trying to understand those. But then what we do in this chapter is we're kind of taking a shift, and that shift is frankly going to last most of the rest of the semester, where while we'll still talk about some proximate mechanisms here and there, we're going to be focusing more on evolutionary or ultimate mechanisms moving forward. So this chapter is about trying to understand how it is that organisms do two main things, and both of these things are fundamental to survival. They're spelled out in the title. So the title is Avoiding Predators and Finding Food, and that is going to be our focus. So I have a little bit of an introduction at the start of this thing, but then basically, again, we're going to spend the whole time talking about these two major topics that are fundamental to the survival of organisms: avoiding predators and then also finding foods. And we'll have finding food, and we'll have several subtopics within each of these categories. But you'll see that there are a couple common things, and we'll sort of get into these with our intro here. So we'll be talking about optimality theory and game theory as sort of two complementary mechanisms. I'm not even sure they're completely different. It sort of depends on who you talk to. That allow us to try to understand these different topics in I think, better detail. All right, so first off, if we're going to be trying to start looking at evolutionary explanations for behaviors, so what is the basis? What is the what is the ultimate explanation? What's the reason why the animals are doing what they're doing from an adaptive standpoint? A lot of times we're going to be relying on questions about adaptation. So our hypotheses often are going to be arguing this behavior that we're seeing is an adaptation, and it's an adaptation because of this and this and this and this. And so we then need to figure out a way that we could test whether or not those hypotheses are correct, because it's not enough to just sort of make some sort of claim, a just so story, sometimes you'll hear them called, that makes some sort of adaptive argument for a behavior or a trait, but doesn't really test it. Like, we need some way to actually test these things, or else we're just kind of spinning yarns and we're proposing hypotheses without ever actually testing them. Now, if you go back to chapter 1, there were two main mechanisms by which we talked about testing adaptive hypotheses. And in this chapter, we're going to be focusing mostly on the first of those, which was to do an experimental approach. So you might remember the main, I think, example we did on the homework that was related to this is we talked about those mobbing studies in gulls. And so there was an experimental test that was done that says, all right, so if mobbing is an anti-predator nest protection behavior, then if we place eggs throughout a nesting site, we should see higher predation outside of the nesting site than in it, because in the nesting area, you should see better protection of those eggs, and therefore they won't be eaten as much. So that's an experimental approach where we're establishing what do we think the benefits are, and also what do we think the costs are of that particular behavior, and then trying to actually assess them and measure them and demonstrate that they are actually present. So what we would do in an experimental approach, usually we're doing some kind of a cost-benefit analysis, and we're trying to assess what are the fitness benefits of a behavior and what are the fitness costs of the behavior. And do note, I'm calling these fitness benefits. So if we're talking about fitness, fitness specifically refers to reproductive success, right? So if you're asking about fitness benefits, you need to explain how it is that the behavior is improving reproductive success for the individual. That could mean that it's specifically something to do with mating behavior or something about the actual act of reproduction. But it could also just mean that it could be related to survivorship. So it generally is the case. Something that improves your survivorship is going to improve your reproductive success, because if you don't survive, you don't get to mate. So in this chapter, we are going to tend to focus on cases where, ultimately we're interested in reproductive success. But for the most part, what we'll be talking about is how is it that animals can survive? How can they find the right food, and how can they avoid getting killed? Because if they do those things, then they'll be in a good situation to actually have an opportunity to have some reproductive success. And similarly, we'll want to look at what the costs are. Most behaviors will have some kind of benefits and some kinds of costs. And so ultimately, those behaviors that are the adaptations, that are the best available trait within our population, those will be the ones that have the largest benefits and the lowest costs. At least the difference between the costs and benefits should be the best. So if we're going to make adaptive explanations, we need to describe the costs and benefits. And one of the reasons this is so helpful is that if we do so, that leads to very specific predictions that we can then go and test. And that's what we'll be trying to do a lot of in this particular chapter. Did anybody remember, aside from the experimental approach, what the other method, I'm not going to go into it now, but what the other method was that we talked about? So we had an experimental method, but we also had another method. That would be often the contrast experimental, but this one was more, instead of looking at one specific species and trying to measure costs and benefits in that species, you can look across species. Yeah, it's the comparative approach was the other major method. And there's a little bit of comparative approach that we'll do in here. There is like that observational versus experimental dichotomy, which is also true, but I was sort of fishing for the comparative approach answer. Okay. again, we're going to focus on this sort of cost-benefit analysis thing. And this feeds into this idea of optimality theory. So in optimality theory, what you're trying to do is identify what are the fitness benefits, what are the fitness costs for different behaviors. And then what you're interested in is supposing that there's a couple different phenotypes that are possible within a population, which of those has the biggest difference between the benefits and the costs. So this is, you can think of this as like the net benefits are the highest. So the idea of net benefits is similar to like net income. So your net income is your income after you've subtracted out all the expenses. And so that's what we're interested in here is what is our net benefits, which would be the benefits after we've subtracted out any costs to it. So this figure, this is sort of a hypothetical schematic that tries to illustrate this idea, but in this figure, there's 4 different phenotypes, and they're just calling them phenotype W, X, Y, and Z. And so you could just imagine maybe these are 4 different behaviors. These are 4 different alternatives that a particular species could pursue. And then what they try to do is that for each of them, they measure what are the costs. So in the case of phenotype W, it has very, very low costs. X has a little bit higher costs, Y has higher costs, and Z has crazy, crazy high costs, right? So we're looking at costs, but we're also trying to look at the benefits of that behavior. So what would be the fitness benefits? And so in the case of W, it has sort of modest benefits. X has quite good benefits. Y has even better benefits. And I don't know, Z is sort of right around where Y is. Maybe it's a little bit worse. But they're both very high in the benefit category. But what we're interested in would be the net benefit, which would be the difference between the costs and the benefits. So in the case of phenotype W, did it again, there's decent, there's very low costs, but there's only modest benefits. So this line here is, kind of only intermediate in size. This would be the net benefits of that particular phenotype, that particular behavior. X has a little bit higher cost, but it has much higher benefits than W did. And so this dotted line here, this gap, the net benefits between these 2 lines is probably the highest amongst all these. Y has really high benefits, but you have to pay a really large cost to this. So I'm sort of imagining like maybe this is being like uber territorial so that all the female, you're the only male around. So effectively you have access to all the females, but maybe you have to pay a really high cost to be uber territorial and scare off all those males. So you get lots of mating opportunities, but you have to spend a ton of energy to get there. Okay? Whereas, maybe over here, you're not as territorial, but you're not getting as many benefits, but you're also not spending nearly as much energy on it. Does that make sense? Z would be kind of like the worst case scenario where you're spending an enormous amount of energy, so your costs are incredibly high, and your benefits aren't even enough to make up for that. So this would be clearly this is a maladaptive behavior where your costs are higher than your benefits at this point. But we're interested in optimality theory. The idea is how can we look at behaviors and try to identify the costs and the benefits, because we'd expect that those behaviors that have the largest net benefit, in this case it'd be X, has the longest dotted line here, the biggest difference between the benefits and the costs. These should be the ones favored by selection. And they should outcompete these other examples because this one is going to maximize your benefits and minimize your costs. So that's the idea with optimality theory, is that we'd expect animals, when they have choices, to pursue those behaviors that allow them to do this. So what we'll try to do, and I'm going to stop at this point, but what we'll try to do is apply those ideas of optimality theory to how animals manage to avoid predators in social contexts, or using cryptic coloration or aposematic coloration. We'll also apply those concepts to try and understand foraging decisions and trying to, this goes into optimal foraging theory. So how is that animals make decisions about what kinds of prey to pursue? And we will have to do that next time because it's time for Reading.